We have dopamine. Dopamine becomes what, guys? Norepinephrine. And norepinephrine becomes what? Epinephrine. These guys are, all three of them are known as what? Catecholamine. Okay? They're known as catecholamine. Catecholamine. Catch. Echolamine. I don't know. Cat. Okay. So there should be an enzyme. We'll label it the same one, number 1. That acts on all of them. And it's known as COMT or monoamine oxidase. Same thing. So COMT is usually peripheral and monoamine is the central. So I'll use, because we're going to talk about depression, I'll use monoamine oxidase. Monoamine oxidase. So the MAO stands for monoamine oxidase. This enzyme takes dopamine and converts it to a soluble metabolite, so it has no longer the properties of dopamine. It is inactive, so it inactivates all these three. The same enzyme will inactivate all three of them. Is it okay with you? Good. And the other enzyme is known as what? COMT, catechol or methyl transferase. If you transfer metal in the M position, you know, that's what it does. Or the the O position, ortho position, it will inactivate all three of them. Regardless, monoamine oxidase will decrease dopamine, correct? Will decrease norepinephrine, true? Will decrease epinephrine. We're not going to talk about epinephrine because it's not found in the CNS. Is that okay? Or I'll just do this. It's not found in the CNS. I'll keep it, but just ignore it because it's primarily produced by adrenal medulla. As far as I know, and you know, adrenal medulla is on top of the kidney, not in the brain. Not in the brain, so it wouldn't affect your behavior or your and your state of consciousness and so forth, right? It wouldn't affect that. Okay. Now, in depression there is low dopamine and there is low norepinephrine. And the third one is serotonin, which we're not going to talk about now. But can you use medication? If you use medication that inhibits monoamine oxidase, it blocks right here and a block here, what will happen to the concentration of norepinephrine and dopamine? It'll increase. The problem in depression was decreased concentration of these. So there are medication, monoamine oxidase inhibitors. They're not a drug of choice for depression. They're the last drug of choice because they're toxic. They're not preferred drugs. You got to stay away from them. You as a clinician, as a psychiatrist, if you have no option, no other things, you will use that. The best drug of choice for depression is serotonin, selective serotonin reuptake inhibitors, SSRI. Okay?
So, serotonin, that's another neurotransmitter.
Yeah.
It's not involved in that.
No. You can call it, but its serotonin is made from tryptophan, different amino acid. These are made from tyrosine. Okay.
We don't use the MAI because of hypertensive crisis?
Yes, sure. And the hypertensive crisis will only occur, or majority cases, if you use tyramine, red wine, red meat, and things, and cheese products and things like that. Okay, but not in any case. So you have to be careful. But now I'm gonna give you a homework. There are 3 MAOI, and you should know their name for Friday. I'll quiz you on Monday. Monday's quiz, actually. There you go. Please know them. Know their names. There are 3 of them, and just to kind of give you a clue, those 3 should start with P-I-T or PIT. That should be their names. Okay. Their first letter of each name. Okay, so in depression, you know, that's what we care for. We want to increase their concentration.
What about medications, doctor?
3 medications, you should know their name for Monday, for Tuesday's quiz. Sorry, I keep saying Monday. Okay, as far as I know, I am done with tyrosine phenylalanine cycle. Those 2 amino acids, how important are they? A whole day. A whole day we spent on just this one cycle, 1 or 2 amino acids. Know which one becomes essential if there's an enzyme deficiency. Know which one becomes toxic if there's an enzyme deficiency. And if these enzyme deficiency exists, what's the name of that disease? What are the conditions and symptoms and signs of that disease? What do you find in urine? What do you find on CT scan? And so forth. You'll be okay. Really, it's not that difficult. Okay. Let's go on to one more crazy amino acid cycle, then we can go home. It's 11 almost, yeah.
For that amino inhibitor, is it toxic? What kind of toxicity?
It will cause hypertension, sweatings, and confusion, and even more, it can lead to psychotic behavior. Yeah.
So it's all mental.
Yeah. You don't use that central version of it. Okay. Now, we talked about tyrosine and phenylalanine amino acids, correct? So, and their metabolism. Next thing we want to talk about is cysteine and homocysteine metabolism and methionine. So, the next category is what? Cysteine, homocysteine, and methionine. Cysteine, homocysteine, and methionine. Okay? Alright. I don't think it's in your book. I was never able to put it because every time this has been the last 4 years, I never put it in the book. I don't know why. Maybe you should do what I do, put it in your head. So you don't depend on the book. I should start making books that have blank pages. Okay, let's do this. So we need something. So there's methionine. I'll use M. Is that okay? Methionine. Methionine can become Sam. Where's Sam?
All the way in the back.
Wow, what is Sam? I miss you.
I'm still here. I'm asking questions.
Yeah? Well, bring some of the reality in front of me. Don't hide it from me. Okay, and I'll tell you what this stands for, okay, guys? This will become Sa. And this will become homocysteine. Homocysteine. Okay, homocysteine. Homocysteine. I want to remove the M in the center. I like to make it this cycle because that's how it's in my head. Okay. This cycle is 1000% on the exam. Now, they might not show you the picture. They'll ask questions in related to this whole cycle. So please, we're going to take it easy a little bit. Okay? I'll slow down if I have to. Homocysteine can become cysteine. 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 And cysteine will become cysteine. Okay? Are we good so far? Okay, this cycle we have to know. Now, there's something that you have to combine with homocysteine for it to become methionine. Okay? And let's start with this. There's an enzyme number 1. It's known as methionine synthase. Enzyme number 1 is known as methionine synthase. Methionine synthase. Methionine synthase. Okay, methionine synthase. Do you remember what's the first amino acid of all polypeptides? Methionine. Methionine. Number 1, mono, methionine. Right? Now, what you need to do is you need to combine a methyl group. A methyl group. Methyl group. So, you can call homocysteine and you attach to it a CH3 methyl group, it's methionine. That's why it's known as methionine. Methylation of homocysteine produces what? Methionine. But where do we get this methyl group from? There you go. And there you go. Let's do that. So there's a little bit more into this. So I'm going to use a different cycle now. So we will have tetra. Let's use first B12. Let's have B12. Vitamin B12. It circulates in this fashion. Why am I so obsessive and compulsive? I have no idea. But I'm going to do it one more time to make a perfect circle. And yeah, perfect egg. You don't like my circle. Whoa, that was big. Okay, so we're gonna do one more circle like this, and that will be tetrahydrofolate. Okay, so here we should have B12. Now we should make some rules regarding these things. B12 is nothing more than a metal carrier. B12 is a vitamin B12. It's a vitamin. Vitamins are known as cofactor or coenzymes. They're never consumed in the reaction, but they help the reaction proceed faster. The reactions depend on them. Okay, so in this case, B12 will keep circulating. So let's say B12 here carries what? A methyl group, CH3. Now the CH3 is brought by who? Tetrahydrofolate. I should use different colors to keep the methyl group in the same. So tetrahydrofolate is a methyl carrier. And what is B12? Methyl carrier. THF stands for tetrahydrofolate. Tetrahydrofolate. Where's Rahaf? Because she will always tell me my eyes hurt, don't use the screen. It hurts my eyes. Okay. And tetrahydrofolate, where do we get tetrahydrofolate from? From folate. Now we're making a little bit large. Folate will become dihydrofolate. There you go. And the reason I put folate in green, because you get it from fruit and vegetables. Folate, you get it from what source? The source of folate is vegetables, green leaf vegetables and fruit, especially vegetables. So think of F, fruit, vegetables. B, beef. The source of B is beef. Beef. B12. You don't eat beef, you should. I don't know, I heard somebody, they said they don't eat beef. You should. We have all these beautiful foods available. Why you gotta, like, deprive yourself?
You can choose.
Yeah, just eat everything. Eat everything. Yeah. Okay, are we okay so far? Now, the other cycles folate, let's ignore them for now, but as long as now. B12 combined with homocysteine, it will donate its methyl group. And homocysteine will become what? Methionine. So in other ways, if this is methionine, it's the same thing as homocysteine plus a CH3. It's the same thing. Without CH3, it's just homocysteine. With CH3, it becomes methionine. So when you add methyl group to homocysteine, it becomes methionine. Are we good? And this is why I didn't do nucleotides before. I'll do nucleotides on Friday because we need this information first. Now, then methionine combines with S-adenosyl. Okay, S-adenosyl. Adenosyl or adenosine triphosphate, doesn't really matter. Okay, so just know that now. Or you can say SA, it combines with SA. And what would you get? SAM, S-adenosyl methionine. S-adenosyl methionine. Really, I don't care for whether you know the name, just as long as you know SAM. It's an additional carrier of methionine. That's all. And then it will become SA, S-adenosyl homocysteine. That's all. S-adenosyl homocysteine. Okay. Now in this process, they will give off a methyl group. They will donate a methyl group. This whole cycle was only one source and one thing: to carry the methyl group, generate the methyl group. Of course, if you need methionine, it can exit the cycle, right, and be incorporated to amino acid sequence. Of course, but nonetheless, this cycle is a source of methionine. How do you make methionine? Methionine needs a precursor known as homocysteine. So your body can make methionine, but what if you have B12 deficiency? What happens to methionine? It becomes what? Essential. Let's use the right terms. It becomes essential. You can no longer make it. If you can't make it, You have to break it and then eat it. I was just rhyming the break part, sorry. If you cannot make it, you have to get it from your diet. So methionine will become essential. B12 deficiency puts you in that risk. Not eating beef puts you at risk. Being vegetarian puts you at risk. Unless you take vitamins, but again, it's not the same. Okay. It always is the best. In moderation. Now, at the end of the day, so we care for this reaction number 1. Please remember this one. Methionine synthase, the enzyme, requires B12. Requires B12 and homocysteine in order to produce methionine. Okay? Is there B12 deficiency? You better it is. We'll discuss it. We'll discuss that disease. There's B12 deficiency. Okay. Now, methyl group.
Why methionine is very important?
Now, methionine is very important because it's a methyl carrier. It's the source of methyl group. Okay? And it carries the methyl group to donate it to DNA and RNA synthesis. But also methionine is needed for amino acid or protein synthesis. So it has a lot of functions. But this function right now in this cycle, you need to know. This methionine can actually leave the cycle and become part of what? Protein. The first nucleotide of, first amino acid of all proteins is methionine. So you need it anyways. So number 1, protein synthesis depends on what? Methionine. Okay? One more thing. In DNA and RNA, we have nucleotides, right? all of these nucleotides needs a methyl group. We'll discuss that tomorrow. What was pure nucleotide? What does it look like? Double ring? Okay, double ring. You've only seen it in this form. But to make it, first, there's a nitrogen donated by glutamine. And then aspartate will come and form half of the ring. And then glutamine will donate one more nitrogen, it will form a ring. And then the methyl right here, this carbon comes from where? From the methionine cycle. So all DNA needs a carbon CH3. Therefore, you need B12 for DNA synthesis, RNA synthesis. But cells that depends on DNA or depends on high division, like RBCs and red blood cells, they'll be affected on this. Non-dividing cells are not affected. So only dividing cells should be affected by this, correct? If you have deficiency of methyl groups, you cannot produce methyl groups or you cannot transport it. Okay, now please understand that. So the methyl group is then donated to what? To DNA and to other molecules as well, but especially DNA and RNA. Let's talk about that, okay? Okay, so that's one. So we'll talk about B12 deficiency. So please write in your notes, we're going to talk about B12 deficiency. And you know why is B12 important now? Now you know. B12 is needed for methionine production, for carrying of methyl group. That's all B12 is for carrying methyl group. It's a methyl group carrier. It's a taxi that carries methyl group, that's all. Without methyl group, we will have less DNA and RNA production. And we will have less methionine produced. Our body depends on them. We need them, especially dividing cells. Okay, so that's one. So we'll talk about B12 deficiency. What if you have folate deficiency? Folate deficiency, so there's an enzyme, we need to memorize that, okay? Dihydrofolate synthase. This enzyme is known as dihydrofolate synthase. And this enzyme is known as dihydrofolate reductase. Okay, reductase. If methyl group is needed for cell division, for DNA reproduction, can you make some connection between division and a disease? Mitosis and a disease? What disease? What disease is caused because of too much cell production? Cancer. So if you can block dihydrofolate reductase, you will call that medication chemotherapeutic agent. Methotrexate. Excellent. Methotrexate. Okay, so let's write that down. Very good. I was going to give it to you as a homework, but it's okay. Methotrexate inhibits this dihydrofolate reductase enzyme. Okay, so it will only act on which cells? Dividing cells. Non-dividing cells should not be affected. Non-dividing cells, not really. Dividing cells are affected. So you need folate for cell division in a sense, especially B12. Especially B12. So that's one thing. But I need to give me two more medication. One that inhibits the synthase enzyme, and one that will inhibit the reductase enzyme. Two more medications. I'll give you bonus 5% on the exam from yesterday. Raise your hand. Don't say it. If you know the medication that will inhibit these two enzymes.
If I raise my hand, I will get the bonus.
Yeah, if you raise your hand and answer the question. Sorry, the second part I forgot. Yeah. 5-fluorouracil? No. Thymidine synthase. That's what it inhibits. That's a different nucleotide. Yeah. No. Anyways, you're on other options. It's antibiotics. Okay, in bacteria, you have two antibiotics, so you should have raised your hand. I didn't see you. You raise your hand like this probably. Is this raising hand, guys?
That's playing with it.
Yeah. Like this. Anyways, time is gone. If anything, I'll give it to Malcolm. So, okay, you answer one and Malcolm will answer one more. Isna, what's the answer? Nope. Malcolm.
I don't know type, I suppose I
Oh.
I admit that.
Yeah. Okay. What's the drug of choice for UTIs? It's a combination. Ciprofloxacin individually, but as a combination. Augmentin, yeah, you can use that too. Okay.
That's bacteria.
What's this combination of drugs? Yeah. Sulfamethoxazole, trimethoprim. The TMP stands for trimethoprim. And SMX, sulfamethoxazole. Sulfa drugs. Sulfa drugs, they inhibit what? Dihydrofolate synthase, sulfamethoxazole synthase, and trimethoprim reductase. Okay, but know that let me give you some extra information. The enzyme dihydrofolate reductase and synthase in human beings are slightly different than bacteria. Okay, they're different than bacteria. So this medication is used in combination. Usually, you will give it in combination. It's always written in sulfa. Whoa. Sulfa methoxazole trimethoprim. Okay, you can use it this way. There you go. But anyhow, that's a combination of medication. So this is a combination used. That means you inhibit both steps. And to inhibit one step is not so good. If you inhibit both steps, the likelihood of not having folate being reduced to tetrahydrofolate is much higher. But there's a thing. In human beings, the enzymes are different than bacteria. Good? So as a result, SMX and TMP acts on bacterial division, but it does not affect human beings so much. So therefore, otherwise it wouldn't be used. Why would you want to use a drug that is toxic to you? You get infection, you use a drug that will start killing your cells. No, it's not a good idea. But methotrexate actually is used as a chemotherapeutic agent. So methotrexate acts on human reductase, whereas trimethoprim acts on bacterial reductase enzymes. So they will have side effects, but just know the difference. SMX TMP is antibiotic, and methotrexate is a chemotherapeutic agent. Okay. All right. So that's one clinical significance, and you need to know this. Please remember this medication. And when I discuss the diseases, you can make conclusion about these medication, what they can produce as a side effect. Which means at the end of the day, we'll have lack of folate or B12. And that's the side effect you should look for. So it will cause B12 deficiency sort of symptoms and folate deficiency like symptoms. Anemia. Very good. Very good. Okay, so now, so we're done with this. Was reaction number 1 is very important, and reaction number 2. Reaction number 2 is it converts homocysteine to cystathionine. Homocysteine to cystathionine. Homocysteine to cystathionine. Okay? And the enzyme should be known as cystathionine synthase. Cystathionine synthase. Cystathionine synthase. Okay? And there's enzyme number 3, which is cysteine synthase or cystathionine lyase. So this will be cystathionine, okay, cystathionine synthase. That's enzyme number 2, synthase. Enzyme number 3, look, enzyme number 2 makes what? Cystathionine. And isn't it appropriate to call it cystathionine synthase? Yeah. Enzyme number 3, what does it do? It breaks it. It converts it to cysteine. So it should be called lyase. Cystathionine lyase. Okay? And you won't have a problem remembering that. It's easy. Both of them require vitamin B6. Vitamin B6. B6. Vitamin B6. Okay? So if you have B6 deficiency, you will have lack of or deficiency of cysteine. If you have B12 deficiency, you will have deficiency of methionine. Make sure you know the difference. Okay? So we're good so far, right? Okay, now let's break it down. What is the big deal?
Do we need to know the enzymes for SAM and SAM?
No. They're transferase, but you really don't have to care for that. So let me clarify that. What do we need to know? We need to know the reaction number 1, which is methionine synthase. We need to know reaction number 2, very important, cystathionine synthase. Maybe lyase as well. That's all. And then of course here, dihydrofolate reductase and dihydrofolate synthase. And then whatever else is required by those, what vitamins are used by them, and what's the reactant, what's the product. Okay, let's move on. Now, there is a disease. Okay, what if you have homocysteine is converted to what? Cystathionine. And cystathionine is converted to what? Cysteine. Okay. What's the enzyme here? Number 1. Cystathionine synthase, right? What if you have deficiency of cystathionine synthase, which is a common cause of this condition? What will happen to cystathionine? Decrease. Cysteine, and cysteine will become essential. Cystathionine is intermediate, so we're not going to care for it. Cystathionine is intermediate, but cysteine is a true amino acid that's used in protein synthesis. So cysteine will become what? Essential. Good. Now, what will happen to homocysteine? Very good. And what would you call that? Homocystinuria. There you go. Very good.
Why doesn't homocysteine get shunted into the methionine pathway?
It can't. Who? Homocysteine? Yeah, it'll get shunted, but it's majority of them is this the preferred pathway. This is the preferred pathway. Yeah.
Why urea? Why not in blood?
Yeah, it will be in blood too, but it's filtered. Because we haven't done kidneys. Now there's the thing, guys, it's a great question. Why would you have these amino acid increase in urine, not in blood? Okay? Every single amino acid and every single glucose molecule and lipids, in terms of fatty acid, are all filtered in the kidney 100%. Okay, because imagine there's a water pipe, like a sprinkler, and it has holes in it, and the water passes through. Whatever water can leak, it will leak. The rest will go through, right? Same thing with amino acids. It's a random thing. So when it reaches the kidney, blood goes through the kidney. Whatever can filter will filter. Whatever is washed away will go. But regardless, everything filters in the kidney. Amino acid. All amino acids will filter. So as a result, it's more likely to build up in the kidney, not in blood. Because if there's extra, what will happen? You get filtered. Anytime there's extra amino acid in the blood, it will filter. But when you have too much, they will not be reabsorbed at the same rate. Yeah.
I hope this is on topic, but
It probably isn't, but I found that like African American or African descent have a higher glomerular filtration rate. Why is that?
Because they tend to have increased blood pressure.
So it's more damaging.
Yeah. If continuously, it's not only, it's actually this, not only African descent, but also the Arabian Peninsula. Yeah. That region. Yeah.
Will that increase the methionine?
Yeah, methionine will increase. Yeah. But methionine activity depends on B12. It's more regulated. In this case, it will mostly accumulate because it should be used this normal pathway. Now, when you have homocysteine, so homocystinuria, let's talk about their symptoms. And that will be a condition, homocystinuria. And that's a disease. Homocystinuria is a disease. Very good. So now, the enzyme is what? How is it inherited? Recessive fashion. Recessive fashion. Recessive fashion. So autosomal recessive. So usually children will have this condition. When they grow, they will have Marfan-like, that's what Mazuza was saying, Marfan-like structure. So they will have tall, long limbs. Okay? And so they will have that. But that's one thing. More than that, homocysteine is known to cause thrombosis. The major problem is homocysteine is prothrombotic. Prothrombotic. So the major issue we have here is thrombosis. And thrombosis can lead to what? Stroke, MI, angina, and other blockages anywhere else causing painful episodes. Also in homocystinuria, nobody, at least I don't know, most of the patients are albinos. Albinism is also part of it. So they will have, usually they're mostly blue eyes and blonde. Doesn't have to be completely white hair like albino, but it's partial. So, okay. So focus on these three things.
Why the album?
I don't know. I just said that. I don't know. Maybe interferes with buildup of tyrosine. Okay, maybe interferes with that, the negative feedback. I don't know exactly. Okay. So look for those things. At the end of the day, in urine you'll find what? Accumulated homocysteine. But focus on the thrombosis part of it, thromboembolism, because it increases clotting factor activation. Most clotting factor and thrombotic factors have cysteine bonds, and that will help them. So they will get activated, and then they will cause increased risk of thrombosis. Okay, that's homocystinuria. It's because of what? What's the enzyme deficient? Cysteine synthase. Cysteine, most common. But could it be caused by cysteine lyase?
Yeah.
Of course. This is severe, and this is mild. Okay. Could it be caused by B6 deficiency?
Yeah.
You better believe it. But B6 is used by the body multiple reaction, so its deficiency is extremely rare. Okay, its deficiency, for whatever reason, if you cannot transport to these cells, they will cause problems. So keep that in mind. Okay, so we discussed this part. Now let's go to, can you have B12 deficiency and folate deficiency? Yeah. Now B12 deficiency and folate deficiency should involve dividing cells. Because the problem is when you have B12 deficiency, that leads to lack of methylation, right? It leads to decreased methylation. Same thing with folate. So they should affect dividing cells mostly dividing cells. Can you give me the name of dividing cells? RBC, skin, epithelial, white blood cells, hair follicles. Very good. All of those cells. Okay. Now, at the end of the day, both of them will give you effect RBCs primarily. The thing is, RBCs don't have mitochondria, therefore they're affected most. Mitochondria, those cells that have mitochondria, they can produce methyl group by other mechanisms as well. But cells that don't have mitochondria, they're 100% dependent on what? On B12 and folate. On B12 and folate. Okay?
So, Doctor, B12 and folate are two different things?
The different vitamins. B12 is vitamin B12, folate is vitamin B, which one? B7.
Oh,
Folate is vitamin B7. They're both vitamins.
when patients have folate as a treatment, medical treatment, it's always in the pamphlet categorized as vitamin B.
Yeah,
It doesn't say which one.
Yeah.
I mean,
Yeah. So vitamin B has different types, and folate is one type of them. Okay. Now let's go with B12. At the end of the day, both of them will cause what? RBC problems. Now RBC synthesis and division needs what? DNA. So in this case, the RBCs don't have the appropriate DNA, and they will never mature. RBC stays immature. Red blood cells will not mature. So the problem is, with lack of B12 and folate, red blood cells cannot mature. Cannot mature. Okay? Now, let's do this. RBCs initially, what's the red blood cells that you know of? What do they look like? Donut shape. Donut shape. Looking sideways, they look like this, right? And looking from top, they look something like this. And they should have something like central pallor, like donut shape. Good? That's what RBCs look like. Now, and they'll look red. Why? Because they carry oxygen. True? What's the difference between immature and mature RBCs? Mature RBC have no organelles. No organelles. No, in capital letter. No, no, no. They're empty sacs. They're empty sacs. They don't have mitochondria. They don't have nucleus. They don't have ribosomes. They don't have endoplasmic reticulum. They don't have lysosome. None of that. They don't have any organelles. They're just a bag that should carry what? Oxygen. So therefore this bag is full of hemoglobin. It's the hemoglobin that carries oxygen. Okay? Now, mature RBCs, guess what? They have a nucleus. Immature. They have a nucleus. Okay, so if they have a nucleus is full of what? DNA, RNA, and ribosomes. And how do they stain? Good job. So immature RBC will appear blue in color. Blue in color. Compared to mature red blood cells. But red blood cells that are immature, they have a lot of organelles within them, and they'll be large, large, large. Okay, so these RBCs are known as macrocytes. Macrocytes. Why? Because they're large compared to the normal mature RBCs. Okay, and the name for them will be what? Erythroblast. Now, if these erythroblasts are large and immature, you can call them what? Megaloblast. Very good. It doesn't mean like a Spanish person, Miguel, and then blasting music. No, megaloblast. Okay. Erythroblast, megaloblast, macrocytes. They're also known as ovulocyte. They're not disc-shaped. They're oval-shaped. Ovulocyte. Nonetheless, all of them has less ability and capacity to carry oxygen. They don't have the same ability to carry oxygen. Good? Are we good with that? So as a result, this will lead to immature RBCs. They cannot get matured. If they can, that means they carry less oxygen, and that will lead to what? Hypoxia. In case of hypoxia, anytime you have hypoxia due to RBCs, it's known as what? Anemia. You can have hypoxia due to lung problems, heart problems, breathing problems, but whenever the cause of hypoxia is RBCs, whether it's the number, shape, or activity, doesn't matter. As long as the cause is RBC, what would you call it? Anemia. So of course this will lead to anemia. Now in this case, if it is anemia, we have to give them a name. This anemia, you can call it what? B12. That's correct. You can call it B12 deficiency anemia, folate deficiency anemia, to be more specific. But generally, both of them categorized together, you can call it what? Megaloblastic anemia. Megaloblastic anemia. Okay, so megaloblastic anemia. And this anemia is caused by who? Megaloblast. Megaloblastic anemia. That's all we care for. When you have B12 deficiency and you have folate deficiency, they're supposed to lead to what? Megaloblastic anemia. So far they're the same, and I'll tell you how they're different, but just give me a chance and hold your horses. Megaloblastic anemia. Now the symptoms of anemia should be what? Fatigue, pallor. What does fatigue mean? You get tired easily in this case, right? Easy fatigability. Okay, what is pallor? Pale skin. Okay. But it depends on the severity. You should always look for these two things. And one more thing, dizziness. Dizziness. If the brain is not getting oxygen, how would you feel? Dizzy. Yeah. Okay. And then eventually can lead to confusion, chest pain, heart attack, blah, blah, blah. It could be lethal. So when you talk about anemia, the normal concentration of RBCs known as hematocrit. What is normal hematocrit? In men, about 37%. In female, 33%. Again, you can have a range up to 47%. Let's say, just for, I don't know exactly, but if it's too much, like 15, 6 is too much. But range, like in the 30s. Less than 30 is anemia. Less than 20 is anemia. And less than 10 is anemia. Less than 30 is mild anemia. Less than 20 is moderate. Less than 10 is severe. So it depends. The symptoms will depend on the severity. How much oxygen are you able to get? If there's enough RBC to give you enough oxygen, you're okay. But if it's not enough, you'll starve. Okay, so whenever you think of anemia, all anemias are different. But in one thing, they're the same in the ability to deliver oxygen. So those symptoms, pallor, fatigue, dizziness, they all should be the same for all anemia. And it depends on what? Severity. In severe anemia, you will have maybe chest pain and maybe confusion and coma because not enough oxygen going to the heart and brain, and they will not function. Okay, so that's general stuff. Now, next thing, in B12 deficiency anemia, you will have further symptoms. Neurological. Neurological symptoms are only found in B12. Brain B12. Folate does not have neurological symptoms. So how do you differentiate between the two? If it has neurological symptoms, it has to be folate. I mean B12. If it does not have it, it's folate. Okay. Specifically, the neurological symptoms are spinal cord problem. Spinal cord problem. And in this case, what do you call the condition when the nervous system is involved? Subacute combined degeneration. What is combined? Meaning one motor, one sensory deficit. So look for decrease muscle strength. Okay. You can call that weakness. It can lead to paresis, partial paralysis. Okay. And then you can have paralysis, complete lack of movement. And the second thing is for sensory loss. Decreased sensation. Now the sensation we're going to talk about is either lack of sensing pain and temperature. This is the most common one. Or not knowing your position in space. Where are you in space? Okay, which is proprioception. In this case, it will be what? Ataxia, right? It will lead to sensory ataxia and lack of or decrease. One of those. As long as the motor has to be there. Motor has to be there, and then you combine the sensory part to it. And then I know you guys probably seen this before, but quickly I'll go over this too. The spinal cord. So you have to memorize this part for those of you who are new. Spinal cord, when you take a cross section of spinal cord, it should look something like this. Butterfly shape. This is what spinal cord should look like. Okay. The area that I will cover gray is known as the gray matter. This area is known as the gray matter. Okay. B12 has no effect on gray matter. It only affects white matter. Can you make a conclusion? What is gray matter made of?
Neurons, unmyelinated axons.
Cell bodies. And white matter?
Myelinated axons.
Axons or myelinated axons. Very good. So therefore, B12 is needed for what then? Myelination. You can make that conclusion. B12 is required for what? Myelination. Myelin sheath, myelin, so for those of you who don't know that, this thing known as myelin is a fat. It's a lipid. It's used for insulation of electric wires in your brain. It's used for insulation of brain neurons or axons. Okay? So B12 is required for myelin. Nobody knows exactly how, but it's needed for myelination of neurons within the spinal cord. So it would affect this area in red. I'll use red for this area. Okay, it'll affect red, and that area is known as the dorsal column. Dorsal column. Write it down, those of you who don't know. Dorsal column. Dorsal column. Red is dorsal column. Dorsal column. Dorsal column. Red is dorsal column. Dorsal column. And the function of dorsal column is to sense your position in space and touch. Position, vibration, and touch. It gives you the ability to sense position, vibration, and touch. So when somebody touches you, dorsal column takes that information. When the train goes by and you vibrate or earthquake, then this tract is responsible for it. This blue area, it's just an area of the spinal cord. This blue area is known as coracospinal tract. Coracospinal tract. Coracospinal tract. Corticospinal tract. And it's responsible for movement, voluntary movement. Okay? And then you will have this area along the border right here. This is known as spinothalamic tract. Spinothalamic. Spinothalamic tract. Spinothalamic tract is responsible for sensing pain and temperature. Pain and temperature. So it depends what is affected. I'll use maybe dark color to see if there's a problem. So B12 leads to deficiency of at least defect in what? Or degeneration of what? Coracospinal tract. So it will lead to weakness, muscle problems, and one of the sensory. Dorsal column is sensory, so is spinothalamic. Let's say these two. Because they're close by, usually affects those two areas. At the end of the day, the neurological symptoms in B12 deficiency should be inability to sense pain and temperature. You know, somebody maybe pinches you or takes a needle or a pin and pricks you with that, and you won't feel anything. Okay? And then of course you will not feel hot and cold. Hot and cold. Okay, and then muscle weakness or paresis or complete paralysis. Okay, so that's different than folate. Folate doesn't have that. Folate doesn't cause these problems. Okay, only B12 deficiency will give you neurological symptoms. Okay, so how will we differentiate based on symptoms? If it has neurological symptoms, we will think it's B12. Both of them should have anemic episodes. Both of them will have anemic episodes. They both cause megaloblastic anemia. But only, so let's do this. Both of them will cause megaloblastic anemia. Fatigue, pallor, dizziness, both of them will cause that. But only B12 will give you neurological symptoms. Now in GI we'll learn that in about soon, that B12 is absorbed in the GI. So if you cannot produce intrinsic factor, then you can have neurological GI problem too. So maybe neurological symptoms and GI symptoms. And the GI symptoms should be diarrhea. Diarrhea, weight loss. Diarrhea and weight loss. Look for those things. Okay, diarrhea and weight loss. So I'll come back to it in GI. How do you absorb B12? That's a different case now. For now, just know that B12 deficiency will give you 3 symptoms. Hematologic and neurological and gastrointestinal. 3 things. So, anemia, ataxia, diarrhea. Whereas folate does not do that. Folate only gives you anemia. Are we good? But we're not done yet. We're not done. Okay. Now, when you take a blood test, so when you want to know if this person is B12 deficiency or folate deficiency, take a sample of blood. Okay? You take a blood test, a sample of blood and plasma. In plasma, if you find there is deficiency of B12, lack of B12, then it's B12 deficiency. If there's decreased folate, then it's folate deficiency. So that's one way. You can also take peripheral blood smear. Peripheral blood smear. That means you take a blood from your veins and you put it on a slide and you look under microscope. Peripheral blood smear. Peripheral because you take it from your veins, not from your bone marrow. Bone marrow will be central. You can take a biopsy of bone marrow, central smear. You can take a smear of your blood from your veins and put it on a slide, look under microscope. It's peripheral, away from the center. So on peripheral smear, you will see, when you put that blood on a slide and you draw it and you look under microscope, what would you see? You will see blood cells. And what kind of blood cells would you see? Megaloblast. You see that if I write this as a part of the question, you'll see megaloblast, sure. Anything else? Ovulocytes, not disc-shaped RBCs. Anything else? Keep that. Blue, there you go. But when I show you a stain, not these words, and then you should see that the RBC should be slightly bluish and large. What if I don't have in the question large RBCs spelled for you? I just show you a picture. And that's what I will do. Make sure you're able to recognize the picture. So go home and search a slide of megaloblastic anemia and look at it. If you have question, see, I forget, I'll look for you one too. Also that, but you know what? White blood cells are also affected, so you will have neutrophils, hypersegmented neutrophils, according to Hani, he's correct. Hypersegmented neutrophils. This is a clinical. This is a huge sign. Look for that. Hypersegmented neutrophils in both cases. When you look at neutrophils, they will have their their nuclei in segments. That means they will have multiple nuclei. Hypersegmented neutrophils. They're immature neutrophils. Immature neutrophils.
But this is not specific to either one.
No, it's general for both.
Oh, this is for both vitamin
For both.
B12 or folate.
Yeah. So far, look. On a blood test, the only thing that will be different is what? B12 deficiency here, folate deficiency here. But megaloblast for both of them, hypersegmented neutrophil for both, and ovulocytes for both. Okay. Anything else? Immature RBCs, yeah. Doesn't matter, all the terms I used before. Yeah.
So the only difference is that B12 just has neurological.
In terms of symptoms, yes. Yeah. Okay. But in terms of peripheral smear, they're identicals. Blood test, no. If you think about serum test, you need to take B12. It will have B12 deficiency, and folate should have folate decrease in blood. Okay. There's a lot more to B12. B12 questions are guaranteed. I believe you will get 4 questions on B12 metabolism, which includes the cycle we discussed earlier. Malcolm. Yeah. Erythropoietin is supposed to increase RBC numbers, so when you have lack of oxygen, the biggest signal for erythropoietin production in the kidney is what? Lack of oxygen, hypoxia. So now any hypoxia will increase erythropoietin. And it's common sense. If you're not getting oxygen, your body thinks, oh my god, this guy might not be having enough RBCs. Let me increase the number of RBCs. You're supposed to, when you have hypoxia, the body compensates by increasing all those things that helps in the transport of oxygen. Heart rate, yes. Heart contraction, yes. Breathing, yes. Bronchodilation, yes. RBC synthesis, yes. Hemoglobin synthesis, yes. And then of course, essentially, increase vascularization as well. So everything that has to do with oxygen transport increases with hypoxia. So to make it easier for oxygen transport, right? And we'll discuss hypoxia. Remind me one day, when you come early in the morning, the first questions, when you ask me, it's great. So ask me those questions, because sometimes I won't cover those things. I expect you guys to know. Hypoxia is a great topic. I have to discuss it. So Friday, remind me. Alright, good. So we're done with the folate, B12, cysteine, methionine, homocysteine cycle. We're done with phenylalanine and tyrosine cycle. Good. The disease related to each. Spend time on them. For Friday, please review nucleotide synthesis, which needs glutamine, aspartate, and glycine amino acids. 3 more amino acids. So for nucleotide synthesis, what do we need to make nucleotides? Let's just not say nucleotide. Let's say nitrogen bases. Okay, for nitrogen bases or nucleotide in that case, we need glutamine, which is amino acid, right? Glutamine should be a donor of what?
Amino.
Yeah, this carries ammonia. So whenever we need amino group, nitrogen, remember nitrogen bases should have what? At least nitrogen. They're called nitrogen bases for a reason. So in order to make them from scratch, we need a source of nitrogen. It's glutamine. Okay, and another source of nitrogen is aspartate. Okay. Now the source of methyl group you also need, so these are both sources of nitrogen. Now also we need a methyl group or a carbon. Where do we get carbon or methyl group from? Tetrahydrofolate. So we need them. Okay. So again, the same methionine cycle will contribute to this as well. Glycine also gives you the first carbon. Glycine will give you the carbon and the amino group. Okay, so glycine is needed, glutamine, aspartate, tetrahydrofolate, in some cases bicarbonate or carbon dioxide as well. Okay, we'll go over this. Huh?
Methionine also or
No. At the end of the day, like when you look at the whole body, let's put it this way. Can you guys live without me? Can I live without you? I'm making a general statement. Can I live without other human beings? No. So we depend on each other. In the body, every reaction is tied to something else. We have to teach in isolation so it's easier. But all the organs work together. And then each organ, whatever happens, works together. So all these reactions are somehow interconnected, of course. Right? Think of your whole body as a city of Chicago. You have everything there.
That's